Good afternoon, everyone. Welcome to the second day of our Gyan course. This is the first hands-on experimental session. So far, we have had four lectures, and in today's hands-on experimental session, you will get to experience all that you have learned through the lectures. So, today's hands-on experimental session actually has been prepared in collaboration with Professor Prasanna and his team. So in this hands-on session, we will actually start from medical imaging 101. So introduction to medical imaging. Then we'll move on to how to handle medical imaging data. So for this, there is a software which you will all get to use. You can check, or at least you will see how this software works in action, 3D slicer. And then we'll look at basics of Radiomics. This is a package that you will be using through the Google Colab. There is an environment, there is a, using your web browser, you can use Google Colab, and Google Colab allows you to access GPU from Google servers and run your own code without having to install any software on your own laptop. And then we look at basics of pathology informatics. And finally, uh, the big chunk or the major part, which is the foundational models. So nowadays, a lot of people are working towards development of foundational models for different applications. So you look at foundational model development in relation to medical imaging. So with this, I hand over to Professor Prasanna for further details.
Thank you. So I'll just quickly to go over some of the topics that Monak will cover in more detail. So in introduction to medical imaging, what we'll have, so we saw how we can apply, we can extract radiomic features from images like MRI scans, or if you remember the lung vasculature extraction that was done on the CT images. We covered a couple slides on ophthalmic imaging, if you remember, so the ultra wide field fluorescent angiography scans, and also for the dental imaging we had CBCT scan. So these are all different kinds of imaging modalities that you will encounter when it comes to working with radiology data. So obviously the most common modality is the X-ray modality or the radiographs. So this is an example of a small cell lung cancer CT scan. So you'll, over the course of this tutorial, which will last all of today and possibly tomorrow, depending on how much we can cover today. I mean, we have other topics that we have lined up tomorrow as well. But you'll learn to load these images. First, let's say that you have a data set. How do you load these images? How do you view these images? So there was a there's a software called 3D Slicer. There are other software like ITK Snap. Uh, I don't think we'll need you to download this software right now. So we already have that on our machine, so you can see how to work with those. So software like 3D Slicer, and Monak will go through it in more detail. So you'll see how you can not just upload images, view images, but these softwares also provide other basic functionalities, such as if you have a tumor, you can upload the mask, or you can overlay the mask on the image and see where the tumor is, where the boundaries are. You can also do some very basic measurements. In fact, you'll also learn about pyradiomics, what we had in the topic slide. So what pyradiomics is essentially a library that you can use to extract radiomic features. So it has a bunch of radiomic features. You can just it's wrapped, it's sort of a wrapper that you can use to call, and then call the functions to extract these different kinds of features. We won't really cover a lot of classification examples today, so those are the ones that we'll keep for tomorrow. But you'll see, how to play with, how to invoke the pre-trained weights and all of that for many different things, along with the foundation models. So I haven't really covered much of what foundation models are. So I mean, foundation models is essentially it's a new terminology. It's something that's thrown around nowadays. All that it means is models that have been trained on lots of data, right? So instead of having models that is very specific to or very localized to your institution, maybe hundreds or thousands of data points, you have millions of data points that these models have been trained on. So those are the foundation models. And it's not necessary that if you download a foundation model off the shelf, it will work for your data. And most likely it will not. It'll work in the sense that it'll give you some results. It might not be the best results. Usually, foundation models are fine-tuned further for your own purposes. So with that, yeah. So these are other kinds of images like PET images. Like I said, we'll look at the tumor segmentations, other objects of interest. We'll also look at pathology images. So if you remember earlier today, we covered digital pathology and how you can open these digital pathology scans. These are huge images, so bear with us. It'll take some time to actually load these images because we don't really have the specialized software for that. You'll have to use, we'll still use Python, right? Yeah, for doing this. And yeah, so with that, I'll hand it over to Moinak. So by the way, for those of you who don't know Moinak, so he's soon to be Dr. Bhattacharya. So he'll be defending. He's in India to get married, but I had him come over because he's he's sort of used to working with this data, and he's aware of some of the latest models that are out there. So I thought it would be a good idea for you to just interact with him. And also beyond this tutorial, feel free to reach out to him. So he's about to start a postdoc right now, and he'll be soon in the academic job market. So he'll be able to also talk to you about what are the prospects out there when it comes to getting jobs in medical image analysis, or staying in academia. He also has some industry experience as well. So yeah, Mana, over to you.
Thank you very much, Professor Veg and Professor Prasanna for the introduction. So uh, let's start with some introduction to medical imaging. I hope you guys have the Google Colab drive or the drive that has all the Google Colab files and the images, right? Okay. So in the file there is one IPython notebook named NITR Session 1 Basics. So if you just download it, I can just give a brief tutorial on how to download this and upload in Google Colab. So it's simple, just download it. It will be downloaded in your downloads folder or whatever folder you have uh, set to your system. For example, I'm downloading it my download folder. You can go to any browser, just type Google Colab and welcome to Colab will pop up. Just click this and you can click this new notebook option. Okay, is it visible now? Okay, so I hope we have a Colab window open. You can go to File, click Upload Notebook, and then click Browse, and then wherever you have downloaded this basics IPython notebook, click upload, and it will take some time depending on the internet, and you will have the Google notebook uploaded. Okay, sure. Okay, sure. Okay, sure. So are we stuck anywhere while uploading the Google Colab? And are you able to upload the basics file? Yeah. Meanwhile, people who were able to log in, uh, because this step takes some time, just click this folder button, this folder, and if you click this, this will restart or this will start your Google Colab session. So something like this will pop up, and click this upload. Oh, meanwhile, also download this chest X-ray image. That also you can do. Sometime what happens is if you are logged in through your institutional email, it might not support Google Colab. But if your institution supports it, then just right click Google Colab and open it. You can click this open with and then open in a new tab. Yes, so download the chest X-ray image uh, and this TCGA A62686 file. Download this, and then once you are here, uh, wherever you have downloaded it, just upload them. So just click that file and click upload. It'll give you a warning, just say OK. And same goes with the TCGA file, just upload it. So the TCGA, this file is a histopathology image. This is a big file, so it might take some time.
First of all, download all the files. Yeah, we have ample amount of time, so we have around 3 hours, so don't worry. And if not, uh, but we don't want you to miss out so that you can actually see how things are working. So
Yeah.
please download the file from the drive link. So after you have downloaded, you can open Google Colab. So if you are logged in into your Gmail, you can simply go to your browser, type Google Colab. It will give you a link, or you can do Google collab.research.google. There is some link. So if you have downloaded, go to your Google Colab and then upload, right? With any specific file they need to upload.
No, so as of now, you can upload the chest X-ray and the TCGA A6 this file. The way to upload the file is click on this up arrow button, hover to the folder that has the files in it, click the
Yes.
files, click upload.
So on the left hand side, you will have a space. Yeah, like that.
So yeah,
Just check bench by bench. If you have downloaded the files, please upload it in your Google Colab window. If you are not able to do it, please just raise your hand. We'll come and help you out.
yeah. These are the critical steps. So if you are not done through this step, we are not moving forward. Test images that will not take much time, but the SVS file will take some time. So if everyone has uploaded the chest X-ray image, we can move forward.
No, just wait, please.
Oh, okay.
Few people are here.
Yeah, sure, sure. Take it.
So the files are a bit large, around 400 MB. You can use your Wi-Fi. Don't use your mobile net. And please download. Once you have downloaded, you can upload in the collab window. We'll wait for some time. So if you are getting that slide, like you can see there are three lines. Sometimes it's not visible. You have to press that. Uh,
Yes.
yeah. Sometimes it's hidden. If you press and then you can press the upload button. Yeah.
Let me start with some basic discussions about what are machine learning, medical imaging, and why do we need this. So the first modality that I'm going to talk about is chest X-ray or X-ray in general. We know, like since our childhood, why an X-ray is needed. It's mainly to check fractured bones, or sometimes pneumonia. Like the first step, if you have like dense accumulation of fluid in your chest. So generally, that's why we need a chest X-ray. A CT scan, it's so chest X-ray. It's a 2D image. So I don't know. Maybe all of you might have gotten chest X-ray at some point in your life. So you will just stand, just like either facing or putting your back towards the receiver. You will have an emitter in the front. The X-rays will come across your body, and the receiver will receive it. So it's a 2D modality. Whereas CT scan is a 3D chest X-ray. I mean, in basic terms, they will make you lie down, and the 3D like the receiver and the emitter will pass through your body. Uh, and MRI is also similar. It's a 3D imaging modality. But can anyone say? I'm assuming most of you are BMI, biotechnology, or whatever in the related fields. What's the basic difference between a CT and an MRI? I mean, very basic. Like I don't think you need a very high level understanding of medical physics. But what's the basic difference of MRI or and CT?
In CT mostly means we are using X-rays, so which gives image based on the different absorption. Means different tissues absorb different wavelengths, and in MRI it's based on the principle of magnetic field, and means there is a strong magnetic field in between V goes, and then a radio frequency pulse will be applied. So its hydrogen atoms will excite, and when we apply radio frequency pulse, it will come down to the normal state, and it gives us what signals.
The K space of that magnetic field will be stored, and then we use that case space to reconstruct the MRI.
Yes, yes,
Exactly.
right.
Thank you. So, and another difference between a CT and an MRI is CT has radiation, whereas MRI does not. So, it's safe to have brain MRI scans brain scans using MRI. Another very interesting modality is ultrasound. It's generally used to uh, uh, do some real-time imaging of the heart or during pregnancies. The only disadvantage of ultrasound is it's very much, the technician dependent. I mean, like a very inexperienced person might get a very low quality ultrasound. Maybe for a radiologist, it will be not of that great diagnostic quality, but a very good technician can take a very great ultrasound, and also again, no radiation. There is another interesting imaging modality. It's called PET. Any idea what the full form of PET? Positron emission tomography. It's mostly used to capture the metabolic and functional part of the imaging, and PET is generally, acquired in conjunction with the CT or MRI. So it's called PET CT or PET MR. So let me go back to the slide, and here I can this is a chest X-ray of the thoracic region. This is a brain MRI where we can clearly see a tumor. Any idea which modality is this? So MRs generally have three, four modalities, the protocols, the sequences. Sorry, the T1 post contrast, the T1, the T2 weighted, and flare. So any idea what modality or what sequence is this? T T1 what? So this has contrast. You see, like there is contrast here. So it's T1 post contrast. This is a lung image of a patient with small cell lung cancer. And this is what I was talking about. This is a PET imaging. The left side is the PET, and the middle one is the CT. And this is the PET CT registered, where like you can see the glucose decomposition happening. So here, and the brain has higher level of uh, glucose decomposition. Uh, Generally for PET, we measure the glucose decomposition using SUV. That's a metric. Okay, so and PET is generally very useful for to detect tumors, or to detect very small tumors which are not very readily visible by naked eyes. Okay, so now we'll work with some libraries which we generally use to upload medical images. Here, I already have written the code to install some Python wrappers, PyDicom, Nibable, OpenCV, Matplotlib, and scikit images. So PyDicom is when we acquire a medical image, we acquire it in DICOM format. So Python created a library which eases our way to upload, download, save an image, and work with DICOM images. We call that PyDicom. And then there is another way of storing medical imaging modalities that's called the Nifty, the NII.gz. And Nibable is another Python library which is very handy to upload and download and work with Nifty files. So to run this Colab cell, you can either click this run button, or you can just press shift return or shift enter, whatever works, and this will download or install all your dependencies. Already done? That should be fairly easy.
Yes,
So
thank
just a
you.
quick suggestion, we are installing all these things in Colab very easily. Don't try to do this in your local system. It will just mess up with your environment. The good practice is if you are working on if you want to run something on your local computer, it can be a Windows or a Mac, doesn't matter. Install Anaconda, create an environment, a Python environment. It's recommended to create an environment with the updated Python version, and then in that do all this experimentation that you want to do. Please don't do it in your local system, or you can use Google Colab. It's very easy. Okay, so uh, we uploaded. I already discussed what SpyDICOM, Naviable, etc. Now, the first step is loading and displaying a medical image. So I have already, I hope the chest X-ray is uploaded. Let me also look at. Yep. And so now I have put the right path here, but the correct way to do is just if you move your cursor to the chest X-ray, you will see this three dots. Click that, go to copy path, and then you can just paste it here. I have already done that, so that's you don't have to worry about it. Now, so what I'm doing here is, given that the chest X-ray image is a 2D image, it's generally stored in a PNG or a JPG format, sometimes stored in DICOM. So if it's stored in JPG or PNG format, we can use a library called OpenCV. So the way to import an OpenCV is import CV2. That's what I have done here. And the way to the code to read a PNG or JPG file using CV2 is cv2.imread. It is image read, as simple as that. And in curly brackets, just put the path, and it will spit out the image. But this is the image array. You can't just, like, if you print uh, image CXR, it will return some numbers. So how a computer stores an image, it stores as an array or a list of list. I don't know what programming languages do you use or what programming languages you have learned. I started with C++, so I call it a list of arrays or an n-dimensional array, but like people in Python like to call it list of lists. Okay, so if you just print the image CXR, you will just see a bunch of numbers. Does not make any sense. But if you print the shape, it will return 629 cross 630. That means there are 629 pixels in the column and 630 in the row. And so how do you visualize these images? You use a library, another Python library called matplotlib, and the correct way to plot a chest X-ray is plt.imso, that is image so, and in that you just put the image CXR as input. Let me quickly remove this and print. You see, this looks like a very, colorful chest X-ray, which is generally not the chest X-ray that we would like to uh, see. So what's the difference? What's happening here? Any idea why this looks like a greenish bluish chest X-ray? Okay, so what happens here is, if you try to plot anything using matplotlib, by default takes into consideration that this is an RGB image. What's an RGB image? Any image that you click, somebody was clicking a photo here. So any image that you click using a camera has three channels, the red, green and blue. So any natural image you see, you're seeing me, you're seeing the board, you're seeing this world. It's basically consists like a combination of these three primary colors. But medical images are grayscale. It's between 0 and 1. So if you want to plot something in matplotlib, you will have to use a simple argument called cmap. Sorry, cmap equals gray. That means whatever you are trying to plot will be plotted as a grayscale image. Now if you rerun this cell, you will see a chest X-ray, which is generally the chest X-ray that we have seen. Any questions? No? Okay. So loading a chest X-ray, a 2D image is easy. Now let's see how to load a DICOM image or a Nifty image. So again, the same upload button, which we are facing so much problem with. Uh, uh, Have you guys downloaded the MRI? This, yeah? Okay, so upload any sequence, any except maybe this 19 KB one. Upload any of the sequence, should also not take that time. Ideally the underscore 00001. And let's see how to upload. Yes, I'll stop. Yeah. And then let's see how to upload a Nifty file using, you know, any of the libraries that we are using. Yeah, I think they'll just check if you are able to upload or not. So I'll just wait. Yeah.
Yeah, I think you have seen CT MRI folders are there. You can open that folders. There are like in
Five
CT there
files.
are 4 files. Yeah,
CT there is one
one
file.
MRI there are 4 files, I guess.
Five files.
Five.
Yeah. Four
So you can
sequences.
open any one of those. All are DICOM .nii.zz format. And then the same way you have uploaded MRI, sorry, the X-ray and the TG slide, the same way.
Any questions on CV2 and Matplotlib? If you are going to work in medical imaging, reading and plotting medical images is the way to go. As I was discussing, if you want to upload a Nifty file, which is basically this nii.gz, you'll have to use this library called nibibble. So the way to import is import nibibble as nib and again matplotlib, just to visualize what we're reading. Now, you remember like for chest X-ray, the command was cv2.imread. Here to load a Nifty file, we'll have to use nib.load. But let me give you some uh, points here. So if you run this code nib.load and just pass the, oh, by the way, same, just click this three dots, click copy path and paste it here. I've already pasted it so that you don't have to do it again. And if you just use this nib.load and image, this will output you something like this. Class nibble.nifty1.nifty image, data shape, fine, and metadata. So when you acquire a medical image in Nifty or in DICOM format, you just don't store the image. You store an entire set of metadata. So what is this metadata? For example, let's say there is a CT scanner here at NIT Rourkela, and they are acquiring your CTs. So we need to know what is the pixel dimension. What is the pixel dimension? Some CTs or some MRs may capture the image with 1 mm thickness. Some MRI, let's say any neighboring institute, let's say some institute in Kolkata, they are acquiring image with a thickness of 1.5 millimeter. Now, if you use Python, read that image, plot it, you won't be able to know what's the thickness unless and until that metadata is stored in this header file. This is called a header file. So you see, here I know this MR slice thickness is 1 cross 1 cross 1 millimeter. That means each voxel that you will get has a thickness or has a volume of 1 millimeter cube. There can be other informations like what's the S row, what's the Y row, etc., etc. What's the affine? We'll not discuss what a nifty affine function is. We'll discuss that when we are trying to save something or download something. But for reading, you don't need the header file. Just to read the image, you don't need any information from the header file. So what we generally do is nib.load and then I directly get the f data. So you see, this was not there here. Here there was no dot get f data. But here to load an FT file, we have this dot get f data. So what it does is from the header, it just gets the image for us. And now if you print the image, you see a bunch of list of lists or a bunch of arrays, which means the image is here now. If you want to print the shape of the image, that means what's the volume of this image, you just have to write image dot shape and then print it. So you see the volume of this MRI is 240 cross 240 cross 155. That means the height and width of the image is 240 cross 240 and there are 155 slices. So I'm just trying to plot a middle slice or in any of one of the middle slices, that is the slide 60th. Again, plot plt.imso image. So any idea what does this mean? Image in square brackets colon comma 60. Array slicing. So you guys might have taken the Python course. Can somebody explain a bit more in detail in the mic so that
Yeah, so actually, what it does is it does the array slicing. Here we have put the first one as colon and second one as colon, which means x and y axis. It will take all the data, and the third one, third column, sorry, the third value in that bracket we have used 60, which means it will start from I think 60 to the last.
no,
Or
so 60 to the last is again 60 colon. So I'm just taking the 60th element.
okay,
Yes. So what I'm doing here is if you have a box, just imagine the MRI as a box. I'm taking the entire, like the height and width of the box, but I'm just slicing it through the middle or the 60th slide. So it will just take that slice and plot it. You see what I'm saying? But if you do, so what array slicing means, so if you do something like this, let's say uh, uh, 30 to 50, or no, I think that will not give any image. Let's say 200. Let's plot this. So you see, like let me to understand the difference. First, you'll have to see the. So this is the entire MRI. You see, there are many black pixels on the top and many black pixels in the bottom. But if you do something like this, 30 or let's say 40, comma colon 180, you see there are some black pixels cut off. So you are cutting it off from the x-axis and also the slice. So why this comes handy is, so you don't, so if you upload this image and feeding this to a machine learning machine learning model, the model is learning many black pixels, which you don't want. You just want to crop the image that has only the MRI and then feed it to a model. So this array slicing or this list of list slicing comes handy. So this is how you plot an MRI. If you want to plot any other slice, you can just change the number, and so this is, I think, plotting right across the neck. And then if you increase the number, like let's say 180, sorry, it was just 155, my bad. So let's say 120, it's trying to plot on top of it. Okay, any questions? So similarly, you can also visualize your CT image. There is another CT image. You can try that out later. Upload it. The process remains the same. You can upload it here using this upload button. Change the path there and then print it. Okay. Step 2 is basic image processing. So in let's say I was talking about scanners. So there is a scanner at NIT Rourkela, let's say in building A. It's acquiring image with 1 cross 1 cross 1 thickness, and there is another CT scanner at building B, which is acquiring CT scan with the slice thickness, let's say 1.5 cross 1.5 cross 1.5. And let's say over 1 year you collected 1000 patients each or 1000 CTs each, and now you want to train a deep learning model with your own institution data. Would you combine this data and train? No. No, generally no, because they are of different thickness. There can be different dose because different patients have different dose levels. For example, a patient uh, mm, who, like, you want to see where the tumors are, you will put a high dose CT. Or person who has recently gone through chemo, you don't want to add any other additional radiation in the body, you will take a low dose CT. There is a dose variance, there is a slice variance. Different scanners will have their own variations. Different institutions will have different scanners. The age of the scanners also matters depending on some studies. So like, multiple factors. But if you want to train an AI model with this, diverse data, the model might get confused. So what's the best approach is you do some data normalization and then feed the data. So some very basic image normalizing step is, let's say, so normalization is similar to any data normalization in statistics. Maybe when we took the statistics 101 course, how to normalize the data. Let's say we have a data x. The way to normalize it's x minus mu by max of x minus min of x. Simple. Same goes for any image. You take the image, calculate the mean of the image, subtract it, and then divide it by the difference of the max and mean of the minimum of the image. And then you plot the normalized uh, image, which does not look different in your eyes, but from a machine learning or from a deep learning perspective, the pixel values have changed. So that is one way. You can just run this, and this will be your normalized image. Now, before the advent of deep learning models, there were some sophisticated edge detection techniques. So, how many of you have heard about Canny filter? No, no, no. Okay. So what a Canny filter does is, uh, so your image is a matrix, right? In mathematical terms, your image is a matrix, and if you multiply your image with another this edge detection matrix, it will plot edges of the image. For example, you see here is a chest X-ray, so there is an edge of the lung, then there is a thorax, and then there are bones. So can you just do some basic image processing and plot the edges? So the CV2, the OpenCV library has a function called Canny. So if you just pass this, you see you will plot the edges of the chest X-ray. Now you can play around with this minimum and maximum thresholds. You can change this. This will change the image. You can bring this to lesser. You will see more dense edges. So depending on what you want to do, you can change these thresholds and plot different edges for a medical image. This comes handy when, let's say, if you have a MRI and if you want to calculate the edge of an MRI for skull stripping, etc., basic pre-processing, this becomes useful there. Any questions? Nope. Okay. So now, sorry, yes.
So in the while calling the Canny method, we are passing 10 and 150. So what was that number?
So those are the minimum and the maximum thresholds of the Canny filter. There is something called Canny filter, which is a matrix, and how much. So basically, let's say your image is somewhere between 0 and 255. Between which pixels you want to put the threshold? Between which pixel range you want to put the threshold? That is basically this value.
So those range, so pixels that will form within that range, they will be retained, or
No,
that.
no, no,
Okay.
no. That is adaptive thresholding. I'm not talking about thresholding. I'm talking about, so thresholding is basically, okay, let me do that. Let's remove this. So let's say gray image equals gray image greater than 150. Wait, still printing the edges. So you see, this is what I was talking about as a thresholding. So all the pixels above the value of 150 are retained. Rest all is not retained. So this can be used for lung segmentation. You see, somehow if you play around with the pixel value, you can segment the lung. But in order to detect the edges, you'll have to use a filter. So what's the basic difference here? Here above the pixel value is 0, sorry 1, and below that pixel value is 0. But what an edge detection algorithm does is it takes another matrix, multiplies it in a convolution manner, and then detect the edges. And those thresholds are basically the thresholds of those filters.
Thank you.
Any more questions? Okay. So, uh, and that is actually a very good application. Let me just take some time and show another application. So, for example, image equals nip dot load. I want to load this brain MRI. Copy path. Sorry, and dot get f data. Plt dot imshow image. Let's say 60. I don't want to plot the axis of. Yep. So this is my brain, right? Now I want to just print a brain mask. So in this image, I just want to print where the brain is. So what I can do is based on this thresholding technique, what we just discussed. You see, you can segment the brain very beautifully using very basic thresholding. Now, if you want to, so you see the brain structure. Yeah. So if I zoom in a bit, or I think this is still a poor quality image. The next step is visualization. After that, I'll discuss another interesting point. Okay, so the next step is visualization workflow. So these are all Python-based visualizations. Why do we need this? Because if our end goal is to develop some machine learning or deep learning or some analysis pipeline. You see what I'm saying? So for that, you need to upload the image in Python and then visualize it. And let's say if you ran some segmentation model, you again would like to see those images, how the segmentation happened and everything. You still need to visualize in Python. That's why Python visualization is needed for informaticians like us. But for radiologists or for clinicians who don't want to write code, there are some visualization techniques. So one of the very basic, or I would say very good visualization platform is 3D Slicer. You don't have to download it or install it in your system. You can do it later, but I have it in my system, so I can show you some basic examples how to open a 3D slicer, load a brain MRI image, and visualize. So this is the 3D slicer icon. Um, and so the way to upload something is you just, so this is my MRI folder. You all have this. I will just take any of the MRI sequence, drag and drop here. This add data into the scene window will pop up. It will show me that okay, you want to try, you are trying to upload this sequence, and that's the volume, and I say okay, and this is the weight. Let me close it because I already had the segmentation open, which I never uploaded. So let me try to upload it, open it once again. So there are multiple other visualization tools as well, 3D Slicer, ITK Snap, etc., etc. Feel free to use whichever you feel comfortable with. Um, yep. So as I was saying, just drag and drop, and this is the volume. So 3D Slicer is basically displaying the volume in three different views. Can anyone say what these three views are? Clinical terminology. Sajai, microphone, please.
Frontal, coronal, and sagittal plane.
Which one is frontal? If you scroll, it will scroll through the different slices of the brain, and of course you can see a big tumor here because this is a patient who had glioma. So this is a very interesting way to visualize your medical images. Now I was talking about brain mask segmentation. So brain segmentation is fine, but let's say you want to segment the different anatomies of the brain. For example, you have the cortical gray matter, basically these black regions, and then there is white matter, and then these things, this black hole-like things are lateral ventricles, etc. So if you want to segment these intricate anatomies of the brain, which you know that human brain are kind of, of course it is very different, but the anatomies are kind of the same. The cortical gray matters will be on the side, then there will be white matter, and then there will be lateral ventricles. You can try to use some edge detection, filters to detect the edges of these anatomies and segment them. Some basic, if you don't want to use deep learning, there had been methods which used edge detection for anatomy segmentation. Okay, so that's why I wanted to first visualize it and then come back to that segmentation discussion with which someone just pointed. Now, so this is the brain MRI, and let's say I have the tumor segmentation of this particular patient. Before that, let me also visualize the other sequences. So this is the T1 contrast, post contrast, and then this is the T1 normal. You can still see the tumor here, but the difference between T1 post and T1 normal is the anatomies are much better visualized in T1 normal. I mean, the medical physicist guys will be better, will be a better person to explain this. I'm not a physicist by training, but still trying to do. This is a flare sequence where the tumor, the SNFH, the SNFH signal, which is basically the extended part of the tumor, is very well visualized. And then finally, this is the T2 weighted. Yep. So these are the 4 different MRI sequences which a radiologist will or a neuroradiologist will ideally need to make a diagnosis. For example, I came there's a patient who came in with headache and something, and we want to get their MRI to check if it's a glioma or not. We'll ideally likely to have like to have these 4 different sequences to make a correct diagnosis of the glioma. Now, if I want to also visualize the tumor segmentation on top of this sequence, so what I'll do is, okay, I think I fast forwarded a bit. So again, go to the same folder, and this is my segmentation file. I'll again drag and drop here, but this time I will not select the volume. I'll select this drop down menu, click segmentation, and then click OK. So now if I zoom in a bit, you see the segmentation is very well visualized on top of the sequence. And these are the different compartments of the tumor. So here, okay. So if you click this I drop down icon and then click segmentation, you will see these 3 labels here. If you want to just look at the enhancing tumor, which is generally ideally a neuroradiologist would like to see, that is this enhancing part, you can just click this eyes just next to the segmentation icon, and if you just close those eyes, those labels will be gone, and you can just see the enhancing part. You can play around with this one, if you want to, let's say, uh, change the opacity. You want to make it less opaque so that you want to see the background of the image. You can change the opacity. You can increase the opacity. If you want to make it a very solid label, you can change the space fill as well. So you can play around with your segmentation mask. Any question? Yes.
So are these DICOM data obtained from a single MRI?
At this what data, sorry?
So the different files that we saw in the directory, that belongs to one patient.
The same patient, yes.
So in a single MRI, we get all sort of data.
All the 4 sequence, yes.
Okay.
So that's what I said. A neuroradiologist, a patient, if comes with a case with glioma, a neuroradiologist would ideally like to have these 4 sequences to make a correct diagnosis.
And all these individual DICOM images are actually formed in the right in several layers. So how currently, today radiologists actually evaluate. They go through that those layers manually. Or
Manually, yeah. As of now, I mean, there is definitely some AI automation going on, but yes, still now the radiologists look at those slices and manually.
These four protocols that you see, T1, pre-contrast, post-contrast, T2, and flare, these are the structural protocol sequences. So there are other functional sequences, fMRI, for example, functional MRI, you have PET scan. So these four are typically available, or these three rather, T1, T2, flare, they're typically available in all machines. So any place you have an MRI scanner, you will be able to get these. So the more specialized scans that are out there, you need more other kinds of machines for that. Yeah. So the reason lot of these machine learning papers that you see out there that are working with brain images, you'll see that there are these for 3 or 4 protocols, is because they are the most commonly available ones. Yeah.
Okay. Any more questions? Okay. So if you don't have any other question, so uh, uh, sorry, that's all about visualization. You can try out ITK Snap. Another thing that initially I decided not to discuss, but given that today morning somebody talked about GDC portal and stuff, okay, I'll just wait till the projector is on. Uh, yeah. So similarly, so these are all imaging modalities, right? And if you and like if you are diagnosed with some sort of cancer, it's not just the imaging data; it's the histopathology data. You also have the genomic data, etc., etc. So all the different visualizations are different. So for example, this is all for radiology images. You can use something called QPath or OpenSlide for histopathology image visualization. Uh, uh, there is a very good open source, I would not say library, but a analysis platform called Xena. I can discuss. Oh, it's here. Okay. So let's start with GDC. I think today morning Professor Prasanna was talking about uh, the TCGA, the TCIA, and the GDC portal. So a very quick suggestion: if you want to work with medical imaging data and specifically cancer data, this portal is from NIH, the National Institute of Health from US. You can download this data from here. You can analyze the data, format the different patients, the different information of different patients. What was the age, what was the gender, at what age this patient was diagnosed, what was the treatment given, everything is in this platform. So if you want to design a machine learning platform with multi modal data, the imaging, the genomics, and the histopathology, it's readily available for multiple cancers. So you see, like in the right, we see the cases by major primary site. So all the different types of cancers are here. So if you are interested in any one particular type of cancer, or interested in working in pan cancer data, you can actually use this for your analysis purposes. So again, a quick glimpse about the TCIA portal. So if you just click the cancer imaging archive, um, so here if you go click access the data, search radiology, just taking some time to load. So here, as I think Dr. Prasanna was mentioning, if you want to design your own cohort, let's say you want to work with lung cancer patients, and lung cancer, not only the primary one, by primary I mean patients who developed only lung cancer as their primary cancer, but also want to work with lung cancer which are metastasized. For example, somebody had brain cancer that got treated, but it metastasized to lung. So you can actually design or build your cohort here. I'll give you a very brief, go through this TCIA portal. It's just loading. Let's see. Meanwhile, we will not be, so the talk is all about medical imaging data. But if you're still interested to work with, as I was mentioning, the genomic data, you can go through this Xena browser. So this is a portal by UCSC, and here it's very interesting. If you are working with genomic data, you can visualize all set of, you can get access to all the different data sets, the breast cancer, all the GDC data sets are also normalized here. You also have access to the pan cancer data, etc. You can quickly play around. You can do multiple things with this portal. Like I can give a small example maybe. So for example, I'm interested in brain cancers mostly. So if I go to the GDC, TCGA, glioblastoma, which is one of the most aggressive form of brain tumor, if I go here, you can get all the copy number data, the gene label, the DNA methylation. Somebody was talking about DNA methylation here, the RNA sequencing data and the different phenotypes, etc., etc. So if you click visualization here, and so this is very important. Like if you just want to look at which genes are expressed in which patients, so select a study to explore. Just click here, GBM, which is glioblastoma, TCGA GBM. Then select your first okay, to first variable. Okay. Now I want to see what is the genomic, the structure. So I know which gene I want to see across these patients. For example, for glioblastoma, the most interesting gene that you would like to see is the IDH1. So I just clicked IDH1, and here I can go to advanced, or let me just keep basic and see the gene expression data. See, okay. And then again I want to see genomic between KI67 or any other, let's say um, TP53, somatic mutation, done. So it's loading. Yeah, so you see this is the entire uh, uh, genomic distribution of the TP53 and IDH1. So, you can see papers who actually, derive their hypothesis from these set of genes and then do analysis on their own data. So just to have this in mind. Uh, Coming back to TCIA, it's still loading, so I'll just keep it there. We'll come back to TCIA again. Um, okay. Any questions regarding visualization? No? So that means two things. Either you understood everything or you don't did not understand anything. Okay. So now basics of pathology informatics. So I hope the TCGA, that SVS file, is uploaded. So in order to visualize uh, the histopathology image, okay, brief introduction about uh, the SVS file. So this SVS file actually contains a histopathology image. Any idea what an histopathology image is? I think we briefly discussed in the lecture, but again reiterating how an histopathology image is acquired. So first step, if we suspect, or if the radiologist or if the oncologist suspect that you have a cancer or if you have a tumor, they'll first do the imaging. Let's say I suspect that somebody is having glioblastoma. I'll first do the MRI. I can see the tumor there, but very important, just detecting they have brain tumor is not enough for treatment. So we need to know what is the subtype of the brain tumor. So if it in glioma, there are 2 or 3 main subtypes. So I just now we saw IDH1. So if the IDH1 mutation is there, that is, any idea what, if a patient has IDH1 mutation, or if it has wild type, what does that mean? Okay, I'm assuming you guys are not from genomics background, but I think if it is IDH1 wild type, it's glioblastoma, and if it's IDH1 mutant, it's either oligodendroglioma or astrocytoma. But how do you came to know about this IDH1 mutation? The imaging can't say that. So what you do is you put a needle through the skull, or there are different techniques, but you do a needle biopsy or an open skull biopsy depending on the patient. Put a needle, get some tumor sample out, slice them, put it under a scanner, and get the histopathology slide. So that is basically for one patient in this SVS file, the histopathology slide. And then I was talking to someone in the morning. The next step after getting the histopathology, the staining and everything that we discussed in the lecture, they take the slide and then they get the RNA sequencing or the DNA sequencing. So basically they get the genomic data from each cell. And that is how you determine if the patient is IDH1 mutant or wild type, or it has TP53 mutation or not, or different types of genomic studies. So the second step is the whole slide image, and the third step is the genomics. Combined all, the oncologist decides what is going to be the treatment. So to visualize a whole slide image, you need to install this tool called OpenSlide, and Python also has an OpenSlide Python, and then Matplotlib and Pillow. So I'll just run it. It might take some time. I hope you will not get any error in this Colab files, because I have checked it and it's kind of error proof unless and until you have messed up with some installations. Uh, then the step 2 is load the SVS image. So again, I have copied the path, and yeah. So you see there are 4 levels of this image. What does this mean? So a histopathology image is it's not like an MRI, which is just like one magnification. So histopathology image has different levels of magnification. So it has either 5x, 10x, 20x, or depending if you have a very costly scanner, 40x magnification. So in 40x magnification, you can see each cell and the distribution of each cell. 20x magnification, you can just see something like this, which, well, I can see the tumor here, but I don't. If I want to know the tumor microenvironment, I would like to look at the 40x magnification. Okay, and the slide dimension is huge. So MRIs, we were dealing with 240 cross 240 cross 155, but histopathology, it's like, you see, I don't know, 1,25,661 and 61,000 something. This is the size. This is the number of pixels of the histopathology image. So it's huge, and that's why the loading and uploading and downloading was taking time. Okay, so I'll just discuss a very basic visualization. You just use the open slide, that is slide.get thumbnail. I just want, so basically I'm compressing this entire big 1,25,000 cross 61,000 to 1,024 cross 1,024. So I'm just downsizing it so that we can visualize. So this patient might just have 2 different, slices there. So these are the 2 different slices. You can read, adjust the heights, and visualize it. And if you want to look at, so here I'm just visualizing the level 0, but if you want to visualize different levels, you can just change this level number and look at different magnifications. Okay, so I would just like to summarize the first step of the basics of medical imaging. Uh, there are few public medical data sets. One is definitely TCIA and TCGA and GDC. But if you want to access other data sets, for example, for brain tumors, you would ideally want to access this. Have you guys heard about the Brat's Lighthouse challenge? Brat's Lighthouse. So Brat's Lighthouse 2025 has multiple challenges. It's for brain tumor segmentation or glioma segmentation. Yeah. Yeah, so there are 11 different tasks: pre and post treatment, adult glioma segmentation, pre-treatment intracranial meningioma. So one primary type of brain tumor is glioma, another type of brain tumor is meningioma, and then pre-radiotherapy, pre and post metastasis, etc. So if you want to train, so where I'm going is, if you want to train a deep learning based segmentation model for tumor segmentation, a good start is the Brat's dataset. So download this. It's somewhere in the range of 30 to 40 GB, which I don't think it's that bad. Download it, visualize the images, and try to run a deep learning model. So these are few data sets which comes very handy if you want to train some basic things to try out as a start. And then from there on, you can move on to much denser deep learning projects. Uh, I'll try to share uh, a Google Doc in the last maybe the last day where you have access to all this, or where you will have the links to access to all these data sets and few basic codes that you can use for pre-processing. So any questions on the basics of medical imaging? Sorry, question. Yeah.
It's a question related to the GDC portal actually. Do you generally use GDC transfer tool and the manifest thing? You add to the card, manifest, and then GDC transfer direct.
Yes.
But there are some images which are like 2 GB, 3 GB, and there are thousands of images.
Yes. Oh, so you don't want to download them in your local computer.
Okay.
You should ideally download them to your server, which has TBs of space.
ठीक है.
I mean, but
Yeah.
the good part is, like, if you know that these are the 10 different patients you want to download, then you can just add the 10 patients, and yes, they will again be 30 to 40 GBs
Yeah.
in total. But I mean,
ठीक है.
SVS files are large, so you can't help it. Okay, so now what we'll try to do is same. Go to the folder and try to download this underscore Radiomics IPython notebook and upload it in Google Colab, similar to what you did to the for the underscore basics file. And for the dataset part, just again click this arrow and just upload the brats glee the all zeros underscore 0000 and this 000 file. So like let me view as a list. So there are 5 different files in the MRI folder. Uh, the patient name, but followed by an underscore 4 zeros underscore 0001 0002 0003 and no underscore, nothing, just .nii.gz. So you just have to upload the first image and this one without any underscore one. Just upload these two nifty files in. Okay, let me try to make the screen a bit bigger. A white background, I have no idea how to do in. Um, how about now? Is it visible now? Okay, wait, yeah, finally I figured out. Okay, so you'll have to just upload the first and the last file. So let me know when everyone has at least done this step, and then I can start. Because as of now, we just discussed how to visualize medical imaging. Here, actually, we are extracting some features which can be used for downstream machine learning and deep learning applications. So this step is very important. Yeah, so just the first and the last. So I'll reiterate it once again to the guys who joined just now. So in the folder, um, you will have another notebook named NITR underscore session one underscore Radiomics. So again, upload it. So upload it to Google Colab and upload. You might also have this MRI folder. Just upload the first and the last file. Similar to the previous exercise, click this upload icon. Just upload the first and the last file. So first step is, just repeating for you, first step is upload this Python notebook, which is there in the folder. Upload this in Google Colab. Similar. File, Upload Notebook, or right click, open Google Colab Notebook. Are you able to upload? And then click this upload sign. You might also have an MRI folder. Upload the first and the last file. In this exercise, we are going to extract something which we call the radiomics features. So why the term radiomics? So anything, any thing that has something to do with medical features is called omics, and if it's extracted from radiology images, MRI, CT, chest X-ray, it's called radiomics. Similarly, if you are trying to extract something from histopathology images, you can call them pathomics. And if you are extracting something from gene data, you'll call it genomics. And if you're working with all of these data combined and trying to do some analysis, you can call it multi-omics model or multi-omics data. Okay, and so radiomics had been very popular. Actually, Dr. Prasanna did a lot of work in radiomics. Like basically, he was one of the starting person who did radiomics and a lot of work. And when radiomics got famous, Harvard group, there is a group in Harvard, AIM, AI in medicine, they made a library public, or they made a Python library called PyRadiomics. So if you have the radiology images, sorry, and the tumor segmentation masks, you can very easily extract these PyRadiomic features. So what are radiomic features? So radiomic features are, okay, I'm sorry, I'll just change the theme to right. Okay. So what are these radiomic features? So they can be shape-based features like what's the maximum diameter, what's the diameter, the perpendicular diameters, what's the volume, what's the shape, etc., etc. And then there are shape-based features, and then there are intensity-based features like the GLCM, the gray label co-occurrence matrices, and different radiomic filters like Hog, uh, uh, Log, Laplacian of Gaussian, etc., etc. So the idea of extracting these features is something like this, that to study, so for example, if you have a solid tumor structure, so what are solid tumors? So any tumor that has some shape are called solid tumors. For example, glioblastoma, we saw that it's a solid structure. But again, tumor in blood, which is just in the blood, there is no solid structure, it's not a solid tumor. So for any solid tumor, you will have a shape. The idea is, can you extract some shape and intensity and texture based features from these solid tumors and make a prediction? So what kind of prediction? Well, the first thing that comes to my mind is, if this is the structure of the tumor, will this patient respond to a certain kind of treatment or not, right? Or if it's given treatment or not given any treatment, how long this patient will survive? So that's called the overall survival analysis. Will this tumor after treatment will recur or not? Multiple types of analysis that you can do. So that's the idea about like why we want to extract these radiomic features. Of course, later, I mean now as we speak, there are much sophisticated deep learning features which can do a much better job, much stable jobs, like much stable predictions. But yes, I mean pyradiomic, before deep learning features, pyradiomic features were very important. It's still important, people do use them, but yeah. So, so what do we need for extracting radiomic features? We need the medical imaging data, we need the segmentation mask, uh, and then finally we'll extract the features and then do some machine learning or statistics. Now, the first step is install PyRadiomics. So you can directly install it from the AIM Harvard GitHub repository, or you can also use something called uh, pip install PyRadiomics don't do it because now I think they have an issue with pip install PyRadiomics because of the NumPy version. So as of now, don't use PyRadiomics. If you are trying to extract radiomic features in your local environment, by local environment I mean similarly install Anaconda, make a Python or a Conda environment and then install PyRadiomics there, you can manually change the NumPy version, or just very simple, use this pip install git https blah 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 pyradiomics.git. So again, similarly run this. By the way, so these are not the only set of radiomic features that people use. You can use these features for a start, but if you are trying to develop a machine learning model based on your data, or if the problem statement is different, you can try to think, that's a good research problem, you can try to think about your own radiomic features, like what other features you can calculate from whatever segmentation masks you have that will make a better prediction. For example, I can think of one of our research idea which deals with not solid tumors but vessels of the lung. So lung vessels, of course, there is a shape, there is structure, intensity, everything. But in order to do some meaningful prediction, vessels are very, irregular in structure. So the first thing that comes to my mind, okay, how long is this vessel? How many branches this vessel has? What's the tortuosity? That means what's the curvedness? What the tortuousness of these vessels are? So these are new type of radiomic features that you can think of if you have a different problem statement. So like from solid tumors to vessels, I thought of this set of features. If you have something else, you can think of some other set of features. Now, given that I hope everybody has this, the two files, the underscore 0000 and the Nifty file uploaded. So I'll tell you what these files are. I think I already visualized. So the underscore 0000, that file is the T1 post contrast sequence, and without without any underscore, that's the segmentation file. So what I'm doing here is I'm reading both of them using another new library called SITK. You can use Nifty as well. I just use SITK because I just wanted to introduce another library. So so you can use Nifty and SITK. Like basically, I'm trying to do two things at the same time: another library and PyRadiomics. Okay. And before Nibble, actually we used to use SITK. That was the only or the most prevalent library for uploading DICOM and Nifty files. Okay, so the command goes something like this: sitk.read image, and then this you read this image that is basically the T1 post contrast sequence, and then sitk dot read image, the segmentation file that becomes our mask. So let me quickly try to import matplotlib as plt, sorry, plt dot imshow. 1 second. Anyway, I don't know what's happening. Let me try to rerun again. There is some error. Okay, the image size is 240 cross 240 cross 155. So we'll see how to extract the radiomic features. So from radiomics, import feature extractor, and then from feature extractor you call this function radiomic feature extractor, and then what you do is you try to binarize the mask. That means these are the different compartments of the tumor. The green is the necrotic core, and then the brownish one is the enhancing tumor, and then this yellow is the uh, PE, which is basically edema. So now what ideally you do is pyridiomics does not take this multi color or multi-label input. So you'll have to binarize this. That means anything that is not 0 is your tumor mask. So if I were to visualize it, let me try it again. Let me try to read that in Nifty. Image is equal to nib. Just try to copy this. Okay, it works. So this is the MRI sequence, and let me try to plot the masks. Let me see. So you see, you can see the three different uh, labels of different grayscales. Now, what I'm trying to do is, you see what happened? From multi-label mask, if I just do anything greater than 0, that means anything that has labels becomes 1. So this is a whole tumor mask, not just compartment wise. And you want to extract the information from each of this from the whole tumor now. So that's what I did here. And then if you just run features is equals extracted.execute and pass the image and mask, it will output this pi radiomic features for you. And you see these has all the different features like uh, shape elongation, flatness, major axis length, maximum 2D diameter, blah blah blah. This has first order statistical values like kurtosis, maximum, mean absolute deviation, mean, etc., etc., and then intensity features like the gray label, co-occurrence metrics, difference variations, the IDM, IDMC1, joint entropy, etc. So these are all the different set of pyradiomic features that you can extract. So this is the feature name and this is the feature value for the whole tumor. Now it's a Python question, nothing to do with medical imaging. So you want to study, as I said in the session before the break, that neuroradiologists, of course, all the tumor like all these different compartments are important, but neuroradiologists are particularly interested in the enhancing tumor. So uh, how would you just take this particular level, which is segment 3, and extract the radiomic features? So what will you change here? So as of now, we did mask equals mask greater than 0. So that means anything that has 1, 2, or 3 value becomes 1, and it looks like this. This is the whole tumor. So just to plot one segmentation label, what should we do? Answer. Let's say the value is 1 something. Mask not equals 1. Greater than 1 means it will include 2 and 3 as well. Right. Okay, let's see first like what are the unique values. Oh, so this I see what's happening. Let's see. Okay, so there is 0, 1, 2, 3. Now if I just want to plot 2, so you see this is the last part. Now if I want to just plot 1, I'll have to rerun it again. It's a bit bad coding on my behalf. So this is the necrotic core, and if I want to print 3, which is basically the segment 3 we already saw. Yep. So this is the enhancing tumor that we want to extract the radiomic features. So I will just rerun this code and wait, what happened? Oh, sorry, my bad. I see. Okay. So when we are doing mass equals mass greater 0, print, wait. Oh, I see. We are changing the image to nifty. My bad. Mask equals 3. Now it should run. Yep. Yep. So now you see this is the radiomic features of that uh, enhancing tumor region. Oh yeah. So what happens is this PyRadiomics library only takes SITK image as input. So the moment I converted it to Nifty, it was showing as an error. So what I just did is rather than, so initially let me comment out the code here. Mask equals mask greater than 0. So if we want to calculate the radiomic feature of the entire whole tumor, we are just doing anything greater than 0, that is 1, 2, and 3 becomes the mask. But now, if we want to calculate uh, the radiomic features of only that particular region that we segmented, that we just saw, we just do mask equals 3. So similarly, if you want to calculate the region, the radiomic feature of another region, just do mask equals 1 or equals 2. So this is how you calculate the radiomic features either of the whole tumor, all of or different segmentation labels of the tumor. Any questions? Yes.
How did we create the segmentation mask DICOM image?
So you are asking like how did we get this?
Yeah.
So the dataset had the segmentation with us, like they provided the segmentation labels. How that was done? It was done in a semi-automated fashion. What is semi-automated? So you train a deep learning model with the MRI scans and the tumor segmentations, and let's say you now have 100 MRI scans from NIT Rourkela, let's say, and then you run the deep learning model or you run the inference of these deep learning models on those images. You get the tumor segmentations. They will have some errors. Send it to a radiologist. The radiologist will manually verify that these voxels are wrong, these voxels should be there, these voxels should not be there, and then finally they will save the segmentations after the correction, and then you can use them to extract the radiomic features. As of now, for this data set, the dataset providers gave us the segmentation labels.
Got it.
Yeah. So that's what I said. If you want to work on your own deep learning based tumor segmentation model, not only for glioma but for meningioma, metastasis, all the different types of brain tumors, you can actually take a look at the Brat's Lighthouse 2025 challenge. So that like has almost like it's a state of the art data set now. Okay. Any more questions? So are we clear with extracting pyradiomics? Because this is very critical. Like if you understand this notebook and if you have the right set of data and if you know how to do some statistical analysis after getting the output, you are as good as to like infer your own outcomes and write a paper maybe with just these set of features. People do that. So this file is very important.
Guys, actually, na, you have to do the same thing, okay? Just don't sit. Just do the same thing, whatever things right now is going on the screen. Actually, you have to do the same thing.
But I will share with you these files again, like with all the modifications and stuff. So yeah, but still keep like
But still,
just
actually, you
for
have to
practice.
do because for
Yeah,
practice also you have to do.
yeah. Don't you
Don't
like you
feel
don't have to
idle,
feel
okay?
left out. But I'll like it's good to get your hands dirty by just like doing basic things like visualization. Simple, right? And the best part is in 2025, if you just write image, Colab will write the entire code for you. You just have to do tab, tab, tab. If you like it, it's fine. Otherwise, just write your own code. Just start writing few lines. It will write the entire line for you. So you guys should feel as confident as a coder now because like when there was no Colab, nothing, people had to manually code. And if there was one error, you will figure out after 10 steps. Here, Colab is by default writing the code for you. So if you know the concepts well, coding is not a problem now. So the same course previously, like 2 3 years back, we used to teach from the very basics of coding, but right now, with Colab and everything is like so integrated with AI. So if you are writing a code in Colab, it's integrated with Gemini because Colab is from Google and Gemini is created by Google. If you are writing code in, let's say, like I use VS Studio. Sorry.
I think one thing are you like you guys also facing some problem, right? Like not imported something. Like there is a package or a library called Radiomics that actually you have to first install.
Oh,
Okay,
so
just type pip install and that name
no,
module
use
name.
this. Are you guys able to install this?
I think
No?
you guys install, right? Those things.
Install, right?
Like some of them are facing same
Oh
problem.
yeah,
So
yeah. Start with this one. Don't use this pip install PyRadiomics because it has some bug. Use this one. You guys use the PyRadiomics, right? Able to extract radiomic features, right? Right? Okay. I was in shock that okay, I skipped the installing radiomics part and moved to okay. So the visualization code is this. Are you able to? Okay, let me try to zoom in a bit so that you can clearly see. Is it visible now? Okay. So this Brat's glioma, all this images is just for one patient. But if you want to download the entire cohort, you can download it from the Brat's Lighthouse Challenge. This is a patient. This is basically the first patient from the Brat's glioma pretreatment tumor segmentation challenge. So many TCIA datasets have the radiology images, either MRI or CT, and the tumor segmentations available. So if you can download them, pre-process them, well, pre-processing can be a bit tricky. It's not difficult. If you can pre-process them very well and use these 10-15 lines of code that I shared with you, extract radiomic features, do some very basic statistics, I think that will be a very good analysis. And if you have a good outcome, like you can clearly see that there are you know difference in patients features, etc., that will be a very good finding for your maybe class projects or master projects or something like that. Mhm. That is here. So this is for the whole tumor. Let me just comment it. But if you want to like print a particular region, like this is the ET, enhancing tumor, you can just use this. I'll just comment this out. And if you are facing any problem with the coding, just rerun the files from the top, because I have a bad habit of naming the same. So for example, here I am changing the mask label, but I am renaming it as mask. So you might get some error because I have this thing like I don't want to name it as mask 1, mask 2, mask 3. So after some iterations, I will forget what was mask 1, what was mask 2, and I'll end up with mask 31 or 32. So I just keep the naming convention same. Okay, so everyone installation done. Everyone can extract Radiomics features. Great. Okay. So what we'll do is for the next 20 minutes, I will briefly touch foundation models. And I think Professor Prasanna will discuss foundation models in detail tomorrow. And okay, so what are foundation models? So, like if you okay, any questions before we move ahead? No, right? Yeah, please.
There are two images. So first I upload triple zero,
Mm-hmm.
and then मतलब sequence is wrong. Upload downside image first upload and
Yes,
second.
that is a problem
This also here.
because this image, you see, this image is the mask
Yes. Yes.
of that particular patient, and this image is the image. So that will be a problem. Great that you asked that question. I think another question would be, is it a problem if we, rather than using 0000, if we use 0001, which is basically like, there are four different sequences here. Underscore 0000 is the T1 post contrast. 0001 is the T1 normal. 0002 is the T2 flare, and 0003 is the T2 weighted. So just for this radiomics experimentation, I extracted the features for T1 post contrast, but the features if you upload 0001 will be different, and the features if you upload 0002 will again be different, and 0003 again will be different. So actually tomorrow I will discuss, so same patient, different MRI sequences, but the same tumor mask, different features. How, and let's say different features means different analysis. So how will you combine these and make a prediction? So that we will discuss tomorrow. As of now, just extracting radiomic features. Okay, so foundation models. So anyone heard the term foundation models? Foundation models. Like if you, I don't know if you guys use LinkedIn or not. If you open LinkedIn and if you follow some groups, all the papers or most of the papers are on designing foundation models, fine tuning foundation models, training foundation models, blah blah blah. So foundation model is a new term. It's basically a very large deep learning network trained on like a lot of data. For example, ChatGPT. I'm pretty sure everyone have heard about ChatGPT, Gemini, etc., etc. ChatGPT is a foundation model. What is that? It's a it's a model trained with the entire data that OpenAI has, which no one knows what is that. So uh, if you have large amount of medical imaging data and train a deep learning model in in a self-supervised manner or a supervised manner, if you have the labeled data, that people are calling nowadays as foundation models. So foundation models seem to be improving. So for example, a model trained on, let's say, just 1,000 chest x-rays versus a model trained on 20,000 chest x-rays, 50,000 MRI, and 10,000 CT scans, everything combined, is performing much better than just a model trained with 10,000 test X-rays. So foundation models have proved to be very, successful in downstream tasks like anything, thoracic disease, like pneumonia classification, tuberculosis classification, tumor subtyping, etc., etc., outcome prediction, etc. So why foundation model is important in radiology? Foundation model is definitely important in the field of AI. Why in radiology? Well, medical data sets are small and expensive. So for example, if you want to train a normal, like a deep learning model, non-medical deep learning model with just car images, you will find like millions of data with just car images. But will you find millions of brain cancer data set or a brain cancer MRI scans? Absolutely not. So in case of medical imaging, training a foundation model becomes important. So you might have 20,000 MRI scans, like, and let's say 70,000 CT chest X-rays and 100,000 CT scans. So combined, this becomes a big enough data set to train a model, which is not possible individually. And another important thing is like medical data is very complex. So if you see like natural images, there is a car, it will be just a car standing on a road. And I'm hoping all the cars will stand on a road. It will be very funny if a car is on water. So most of the cars will be on road. But medical data is very complex. It has different anatomies, different diseases. For example, brain can have Alzheimer's, brain MRI can have tumors, brain MRI can have hemorrhages. CT scans can be taken for pneumonia. CT scan can be taken in the same region for cancer. So medical data is very complex. So training a foundation model for medical imaging data is also very different. And there are very few labeled samples. So, if you ask a radiologist here, we have 1 million data annotated, they'll just be very mad. We don't want. And then faster convergence and more stable training. That's of course like the only critical thing in medical imaging is we need best accuracy. We can't compromise with accuracy. You have to be 100% accurate. I mean, almost near 100% accurate or at the always, because like you can't say, well, this patient will die with a probability of 60%. That's very bad. You'll have to be very, accurate. Okay, so as of now, the Google Colab files that we used, we did not use GPU. But now, to even run these foundation models, we need GPUs. So how to turn on a GPU in a Google Colab notebook? So I'm going very slow here, just follow the steps. Click this runtime thing. Oh, okay. So click this runtime. I hope everybody can find the runtime. Then go to this change runtime type. Okay, click that. Here I am hoping your option will be like the CPU. Change it to the T4 GPU. Is it already there? Because I sent you the IPython notebook. So, but still, it's better to be sure that everybody has the GPU, and then save. Okay. So we all have GPUs now. So like we are calling foundation like this, deep learning models trained with large data foundation models now, but like they were always there, like since 2019 maybe. So NIH, National Institute of Health in US, has made a chest X-ray dataset public, which has 14 disease labels, and I think they have almost 130K data, maybe more. I don't remember exactly. And then they have trained a DenseNet. So do you guys know what a DenseNet is? DenseNet? No, no. Okay, then let me try to give a very brief basic about what a deep learning neural network is. So, like, how to explain? The first neural network, I mean, we all hear the term AI now, but AI in some different form was there in 1946. In Cornell University, there was a lab, Cornell Aeronautical Lab, where Professor Rosenblatt discovered a one layer neural network which is now called the Rosenblatt's perceptron. It was just one layer, and you feed in something, it will output a data. So he basically used that for I think character recognition. So he gave handwritten 7, handwritten 8, 1, and that predicted that, well, this is the text which is handwritten is now 1. So that was the first neural network. It's now called Rosenblatt's perceptron. And then there were multiple models. I would say the first deep neural network was AlexNet. I mean, maybe there were a few models before that, but AlexNet became very famous. And then following AlexNet, the big boom happened in AI when ImageNet dataset was made public. So ImageNet dataset is still very famous. It's like has a lot of images, and that basically favored the way of deep learning training. And that is where the convolutional neural network started to enter the domain. So what is convolution? You have an image which is like a 2 cross or like, let's say, height cross width, like a 240 cross 240 image. You take a convolutional filter, which is again a matrix, and multiply that for the entire image, and then encode the features. So that is a convolutional neural network. AlexNet was a convolutional neural network. Now what happened in the year 2016, I just started undergrad, and this paper came in. The paper name was, basically the model name was ResNet. So ResNet, I mean, till that time, convolutional, yeah, it's perfect, it's working accurately, blah blah blah. But ResNet, what it did was, I can just show you the ResNet structure. So this is a figure from the ResNet paper. So this is a convolutional layer. There is another convolutional layer. X is the input. If you just remove this identity part, that was AlexNet. But what ResNet did is it just added a residual network. So the output, rather than becoming f of x, which is a function of x, and function is basically the layers, it becomes x plus f of x, and that changed the domain. ResNet, a residual network, a network with a residual connection, was able to perform very accurately on multiple different tasks. So that was one, like change that happened in AI. And then following that, in the year 2017, another paper came, which is called DenseNet. So what it did is, I'll just show you the structure. Yeah. So they were like, okay, why will we add one residual connection? We will add multiple residual connections. So this becomes a dense network, like electric wires. If you see, I don't know if they're now, but like in my village there is one pole, multiple electric wires coming, and no one knows where it's going. So dense net, and that outperformed ResNet. So for a long time, DenseNet was one of the state-of-the-art method. By state-of-the-art method is it outperformed all the other existing methods for multiple different tasks. So coming back, so uh, DenseNet was trained on this NIH X-ray 14 dataset, and it was made public by Torch Vision, which is a model by PyTorch. So you don't have to train anything now. You can just download the weights and use this for your inference. So now, for example, you ran this code and you have this model. Now, let's say you have an image. You got an image from the neighboring hospital, and you want to see what's your this pre-trained DenseNet performance on this particular image. So I'm just randomly creating an image. This is a random function, uh, and then if I run this, wait, I'll have to print the output as well. So this is the output. This is the output feature from the foundation model. So what to do with this, we'll discuss next day. But this is how you download a foundation model. If you have a chest X-ray, extract the features, and then from there on you can extract multiple different downstream tasks. You can do multiple different downstream tasks. But the objective of this session is, this is how you download a foundation model. You don't have to train one. You can just download existing foundation models. Similarly, there was another function or another library called the Torch X-ray Vision. they trained on, so not only the NIH 14, the chest X-ray 14 dataset, but also other big chest X-ray datasets. The CheckSpurt, the Mimic CXR, the PAD chest, these are all like big datasets in itself. So overall, this become a huge dataset, and they again trained DenseNet on this all of these datasets combined. And you can again download this very easily in Google Colab. So what I'm trying to show is you can download all these foundation models here. Then so these are already there in these GitHub repositories for chest X-ray vision or torch vision. But now, if you want to install something from Hugging Face, uh, so this will throw an error, hopefully. So how many of you have heard about Hugging Face? Have heard about Hugging Face? Okay. So why do you think this error came? So there is an error here, right? So why do you think when I'm trying to download this model BioVIL from Microsoft, it throws an error like this? Just read the error. I think you will.
I think we haven't added our token, Hugging Face token, we haven't added to this.
Exactly. So what you'll have to do is you'll have to create an account in Hugging Face. So Hugging Face is basically the name Hugging Face, the smiley. So you create an account. I already have one. You go to settings. Oh, I'm being fast. Sorry. You don't have to do it now, but just like take a look. You can figure it out on your own. Just create a account in Hugging Face. Go to settings, and then here you will get your access tokens. I don't remember my password. Anyway, so from here you can get your Hugging Face tokens, and then if you upload it here, it will allow you to download the foundation models. If you are really interested in using these foundation models, I can, find out what my password is, come tomorrow and then download this. Anyway, uh, similar for MedClip. MedClip demo I'll show tomorrow because I think we are running short of time. MedClip and MedSam. So Sam, how many of you have heard about Sam? Sam was the segment anything model made public by Facebook, and that was adapted for medical image segmentation. No? Okay, so tomorrow we will discuss MedSam. It became very famous overnight because if you send a medical image, something like this, and if you just like put a bounding box, it was able to segment tumors really accurately. Just imagine. The radiologists now don't have to segment pixel by pixel. They just draw a bounding box like this, and the segmentations are there. And you can extract radiomic features and do so many things. So this became very, very famous overnight. Okay, so the final thing that I will discuss today, because somebody mentioned Monai, I think you mentioned Monai. So yes, so Monai is a very, like, I would say very user-friendly uh, uh, tool to either input medical image or train models using medical imaging, because Monai has made all the models, for example SWIN, VIT, I'll discuss that later, all the models that you need public, so you don't have to write code, just import them and run your own foundation model. So for bioinformaticians or biomedical informatics students like you, you don't have to write the code explicitly now. Your task is to run the inference and work on, like, the results mostly. So if you are using Monai, that basically simplifies your life. You don't have to deal with much, back-end coding. So simple Monai installation is very simple. Pip install Monai, Nibable and INOPS. INOPS is basically Einstein operations, which are basically just simple matrix multiplications and stuff made faster. So if you run that, these libraries will be imported. And then, as I was saying, for example, if you want to write the code of Swin Unitar, 500 lines. But if you just use monai.networks.net import Swin Unitar, 1 line. So that makes your life very easy. and then some basic pre-processing like intensity scaling. We discussed intensity, why we need to scale the intensity, and resizing. So I will load. You don't have to do this because this you can try out um, in the in your own time. It's fairly simple. The CT. So I uploaded the CT image that I had. We'll use this CT image again tomorrow. So don't delete these files, please. Okay, it's still uploading. So yeah, so you see what I was saying, like Monai has already trained the Swin Unitar with a large data set and has made the weights public to you, and you can basically just download the weights and start using these foundation models, which we'll see now. So my image is loaded. My image shape is 302 cross 302 cross 276. Now I will do few basic normalization steps, that is the intensity normalization. So somebody told me about the Hounsfield unit, right? I think you? No. So basically you like described how the C2 is acquired and different organs have different absorption, right? So in CT, exactly, I wanted to like bring that here. Uh, uh, so basically different organs will have different Hounsfield units. For example, if you see in a CT, the bones will be very white and shiny, whereas you can't even see the fluid, the blood, unless and until it's contrast enhanced. And the soft tissues and the organs will have different like images. You have pixel values. In CTs, you have Hounsfield units. So for example, here you took a very high dose CT, so the Hounsfield unit will be very, or the intensity will be very different, and a low dose CT will have intensity of a very different scale. Again, to train a foundation model or even get the inference, the features, you need to normalize it. So what I'm doing here is from minus 1000 to 400, I'm normalizing it between 0 and 1. There is no information loss here. It's just like I'm normalizing it. I'm not like cropping out all the other values. So anything that is below minus 1000 and above 400, I'm not deleting them. I'm just normalizing it. So it's not information loss. And then I'm resizing it again. Like here, my image size is 302 cross 302. There will be another image which will be 512 cross 512. Deep learning models can't handle this, like different image sizes. So the best idea is to just resize everything to 96 cross 96 cross 96. Yes, here there is information loss, no doubt, but that you will have to, you know, deal with. So now my final image is 96 cross 96 cross 96, and then I download the weights. So this weights Monai has already provided us, so it's very easy. And then I declare or I initiate the model here, upload the weights in this. So torch.load basically uploads the load in the the weights in the model, and this is basically the entire model structure, which you don't need to know what a Swain unitary is as of now. And then you just feed in the CT here, and it outputs a feature of say 1 cross 768 cross 3 cross 3 cross 3. So what to do with this shape we will discuss tomorrow, but today I just briefly discussed these are all the foundation models that you would like to use. These are all the features you can extract. Very easy. Foundation model is not something that you can't use. It's very easy to use. You see just 2-3 lines of code and you can do multiple downstream tasks using this.